Well, the answer is because transmembrane proteins have predominantly hydrophobic amino acids. That is because the phospholipid bilayer is hydrophobic in nature. So in order for the protein to cross the membrane and to be embedded inside the lipid bilayer, it must also have these hydrophobic amino acids. And with that, they can interact with one another by the van der Waals forces. Well, for the protein, they are exposed to the environment or to the cytoplasm. They are having mostly hydrophilic amino acids so that they can readily interact with the water molecules, which is bipolar in nature. So for more information, you should refer to Cell Biology Lecture 2. Okay, here is something extra. We can actually predict the presence of transmembrane proteins by looking at their amino acid sequences. So showing here is an online server. What it does is to examine the protein sequence and to see if there is any hydrophobic amino acids, and then to compare this sequence to other known sequences of other transmembrane proteins. So here is the outcome. We can see that the number of predicted transmembrane helix turns out to be 1. And so for this protein, it has 569 amino acid residues. But for residue number 1 to 336, they are predicted to be lying outside of the cell membrane, while the transmembrane section will be in the middle. While for the rest, which is residue 360 to residue 569, they are residing inside or facing the cytoplasm of the cell. So here is just one example how we can apply the knowledge in biochemistry in predicting the presence of transmembrane protein.